Yo, what is up, all my peoples? Okay, it is January 19th, 2022. Okay, the year is starting off strong, and for me, I'm winding up my basics on the immune system video set take one. Okay, we're almost close to the end. Tomorrow, I will be discussing the lymphatic system because what video on the immune system would be complete without talking about where a large portion of the lymphocytes spend most of their time, which would be in your your lymph nodes, which are your lymphatic tissue, secondary lymphatic tissue, um, and your lymph ducts, and you know, in your blood, obviously. But we'll talk about that tomorrow. Today, I want to talk about the immune system's, I guess, one of the most important components of the immune system, and the innate and adaptive immune system both have analogs for this component, and that would be the natural killer cells. Okay, so a lot of people think about the lymphocytes, the standard lymphocytes being like the T lymphocytes, the helper cells, the killer cells, the CD8 positive and the CD4 positive cells, those that are part of the adaptive immune system that kill specific antigens, that kill pathogens that express specific antigens, and which learn this through the interaction of the major histocompatibility complex type 2 in the lymph nodes from antigen presenting cells like the macrophages, things like that, and dendritic cells, etc., things derived from monocytes. So natural killer cells are part of what is called your immunosurveillance system. So what is immunosurveillance? Immunosurveillance is basically the part of your innate immune system which kind of, your adaptive immune system, it also participates in immunosurveillance. So it's generally part of your immune system where there are monitors. There's cells like roaming about looking for abnormalities. For example, if the cells float and they kind of travel through your bloodstream, they happen to run into a cell, a virulent cell. Okay, they will then attack that cell. Right, so they're kind of like patrolling guards or border patrol or something like that, right? So these natural killer cells can be thought of in that way. Okay. Now, natural killer cells and killer T cells, they're both similar. They both have similar attack stratagems, except natural killer cells have a non-specific attacking process / pattern, while as killer T cells have a specific, the antigen detection mechanism, as is all cells in the adaptive immune system equipped with, right? So your humoral immunity, etc. So think about it this way, so natural killer cells, and you need natural killer cells because remember, your adaptive immunity, adaptive immunity kicks in later on during the course of your infection, after your B lymphocytes have differentiated into plasma cells, after enough of them have produced memory cells, as soon as your system starts producing antibodies. And this takes a little bit of time, and it also takes time for your thymus to recruit and it depends on if you get older, to recruit and differentiate T cells, right? Like you have undifferentiated T cells going around. You need the T cells to actually interact with the antigen that's being presented, and then you need them to then differentiate into CD8 cells, okay? So the thing is, right, natural killer cells have a different way of detecting, and again, they detect pathogens, or they detect abnormalities without looking for antigens present on the surface, right, expressed through the major histocompatibility complex. And this is important because not everything once a cell is infected, right, like with a virus or whatnot. Not first of all, not everything in your system has an MHC, right? Pathogens don't have major histocompatibility complexes. It's a human um, it's a human thing, right? Like it's a molecule for human cells, or it's a molecule complex for the membranes of human cells. So what if the natural killer cell doesn't detect those? The T cells can't detect those, so the natural killer cells won't detect those. But the natural killer cells have a dual receptor system instead that detects that essentially has a differencing mechanism. It's actually interesting how it works. There's a inhibitory signal, and then there's an activation signal receptor. So basically, your if your cells, right, have an activation uh, have activation antigens presented on them, right? Strange antigens that the natural killer cell is generalized to detect, right? Then this will increase the number of activation signals. But if your cell happens to have self-antigen, your and each natural killer cell will detect the self-antigen. So if there's more self-antigen, essentially, than there are pathogenic antigens, as indicated in this diagram here, roughly, what's going to happen is the natural killer cell, if there's more pathogenic antigens, natural killer cell will be activated, and once it's activated, then it will perform the killing function. Okay, so this is important because the activation, the killing function is so now when you think of the killing function of cells, you think of, for example, macrophages, which ingest cells, and then you think of your complement system, right? Don't forget about the complement system. The complement system has a C-class proteins and protein complexes which combine on the membrane of different pathogenic cells and pathogens to form the membrane attack complex. And remember, the complement system is a very old and very, in a way, unintelligent system. It's a chemically activated system, so it just reacts completely to chemical signals, and it will form the membrane attack complex on a cell and attempt to lyse the cell. So it'll try to create a pressure differential in the cell. So in a way, this is actually not the best idea for all pathogenic cells. Okay, I mean, for certain bacteria, sure, it's great, because the bacteria simply need to be lysed, and the pressure needs to make them explode. Okay, but this is not always the best option. For example, what if a cell is infected with viruses? What if you cause the osmotic pressure within a cell to burst open? Viruses need that to happen, right? Viruses need to escape because the viruses use the insides, internal mechanisms of a cell to replicate, right? The same way we talked about how HIV replicates in a cell, other viruses also replicate within a cell. And how they replicate, different mechanisms. If they're RNA viruses or reverse transcription viruses, whatever they are, they need to replicate in the cell, and if the cell is busted open, then they escape and go infect other cells. So this complement attack process through the membrane attack complex is not always the best mechanism. So that's where killer cells come in. So killer cells actually have granules. They're granulocytes, right? And these granules have chemicals stored in them called perforins, which comes from the word perforate. So these granules, these perforins, they will go inside the cancer cell in this case, or they'll go inside some other cell, right? Or that's basically has a activation that has activated the natural killer cell, and they will essentially release. They will lock on. There's receptors for these perforins to lock on to the membranes of a cell. They'll lock onto the membranes of the cell, and what they will do is inside the cell, once they lock on, they will activate through a very complex series of chemical intermediates. They activate the caspase cascade. Okay, so what is caspase? Caspase is an important molecule. Okay, let's go. Caspase, and this is essentially something that's important because caspase is basically the internal inflammation mediator. Okay, so caspase within a cell, what it does, it goes around once it's activated and cleaves different pieces of the cell. Okay, and caspase is what once it's activated, eventually they lead to the controlled death of the cell through apoptosis. Okay, so this is important because it doesn't create a dumb lysis of the cell, which causes it to explode and potentially release bacteria. Caspase activation actually kills the bacteria within a cell, and then the cell dies, right? And then basically the once that happens, that's the natural killer cell has done its job, and it can move on without having to worry about having infected the rest of your system by viral infection. So just the one thing, we'll talk about caspases in the future, but caspase activation is actually really important because caspase and caspases have a very particular series of like components. Caspases essentially are created through the interaction of this thing called the inflammasome, which we've talked about before. But these caspases can be activated from an external stimulus, which is how natural killer cells do their killing, or they can be activated internally from within a cell. So a cell can cause its own death, right? And a cell will cause its own death under certain conditions. Like, for example, when we go into extending human life, cells can detect whether they have the potential to become cancerous or not. And if they do, then they will kill themselves, right? They'll commit suicide. So it can be a homicide, a cell homicide, or a cell suicide factor. A caspase is basically a bullet. Okay? Yeah. So, and then that's basically it for the mechanism of natural killer cells. Just to recap on what T cells, killer T cells, just know the difference. Killer T cells do something slightly different in that they detect through their T cell receptor binding, they detect major histocompatibility complex antigen irregularities. Okay, and once they do that, then they will release perforin. So they kill in the same way, but remember, they're geared towards specific antigens. While natural killer cells, their activation / inhibition pathway that activates the caspase cascade pathway has different is non-specific. Therefore, it's needed in immune response. And if you happen to look at the response curves for natural killer cell activation, natural killer cells are activated, they're most active during the initial phase, the latent phase of an infection, right? So they start to build up as the infection builds up, and then they cause kind of the infection, or they're typically indicated in the control of an infection to prevent the curve from going exponential, right? And then the adaptive immune system has time to come in and do its thing later on, following in the 2 weeks of the standard type of infection. So that's all I wanted to talk about. That was a pretty involved video on an involved subject, but I just wanted to tie this part of the immune system up. And yeah, tomorrow we'll talk about the lymph system. All right, have a good night.